All right, are there any questions? So last time we had basically come, I think, to the previous slide, right, where we talked about the role of the equilibrium constant, and that if the equilibrium constant is larger, then the reaction will proceed strongly in the forward direction. Now, enzymes are powerful and highly specific catalysts. Each enzyme will catalyze only one very specific chemical reaction in the cell. And enzymes catalyzing similar reactions, they're grouped together and they have special names that are given to them. Right. Hydrolases are enzymes which catalyze a hydrolytic cleavage reaction. Like we talked about the formation of polysaccharides, right? From simple sugars, right? I mean, by the elimination of a water molecule, that's a condensation reaction. The reverse reaction is hydrolysis. The enzymes that carry out the reverse reaction, that is hydrolysis, they are called hydrolases. Nucleases are enzymes which can break down nucleic acids by hydrolyzing the phosphodiester bond between nucleotides, right? We looked at these nucleotides like AGCT, right, connected to a sugar phosphate backbone, right? Remember the 5-carbon sugar, either like a ribose or a deoxyribose, and then there was a phosphate added, okay? And then we've also seen how those molecules are linked together, right, to produce nucleic acids. So nucleases are basically enzymes which degrade nucleic acids. They will cut those phosphodiester bonds and they will bring out the individual nucleotides. Proteases are enzymes that break down the proteins by hydrolyzing the peptide bonds between amino acids, right? We have already seen that if you take 2 amino acids, you link them together with a peptide bond, right? That's how you get a protein, right? Many amino acids linked together. So if you want to cut these bonds, then you need the help of an enzyme, and these enzymes are called proteases, right? Synthases or synthetases is a general name for enzymes which synthesize molecules in the anabolic reactions or biosynthetic reactions by condensing 2 smaller molecules together. Isomerases. These are enzymes which will catalyze the rearrangement of bonds within a single molecule. Remember, I talked about in organic chemistry, you have several instances of isomers. You have the same molecular formula but different structural formula with completely different properties, right? So how do you go from one isomer to another? That is facilitated by an enzyme that would be called an isomerase, right? Polymerases are enzymes that catalyze the polymerization reactions such as the synthesis of DNA and RNA, right? Because to form DNA or RNA from the individual nucleotide units, you have to link them together by phosphodiester bonds. Okay, this is an energetically unfavorable reaction, and you need enzymes for that, and they are called polymerases. Yes, sir. Go ahead.
Synthesis also
Synthesis.
process is managed, and polymers is also synthesis.
Yeah, it is.
Similar.
Yeah, they are similar. Yeah. Okay. But it may not, like synthase, you might have what they call ATP synthase, okay? That's not a polymerization reaction, okay? That's something that produces ATP from ADP. Adenosine diphosphate, add a phosphate group onto it, you can get adenosine triphosphate, right? So that would be catalyzed by a synthase, and that would not be a polymerase, okay? Polymerase is DNA polymerase, RNA polymerase. They form DNA and RNA from individual nucleotide units. Okay, but you're right, yeah, I mean, polymerase also synthesizes something, so that is also a synthase, yes. Okay, now we come to 2 other groups of enzymes, all right, which are very, very important in eukaryotic cells. Kinases, all right, are enzymes that will catalyze the addition of phosphate groups to molecules, right? So kinase always is going to add a phosphate group, right? Phosphatase will go and remove a phosphate group, right? In eukaryotic cells, the activity of genes can be turned on and off by adding a phosphate group to a particular protein or taking a phosphate group off from a particular protein, all right? So that's why kinases and phosphatases, they play a very important role in eukaryotic cells. In fact, in our cells, the control of cell division is basically carried out by kinases and phosphatases, okay? There are different kinases that will add a phosphate group to something and that will get the reaction started. Another name is oxidoreductases, right? Another class of enzymes is a general name for enzymes which catalyze reactions in which one molecule is oxidized while the other is reduced. Enzymes of this type are often called oxidases, right? Or reductases, or even dehydrogenases. Dehydrogenase means it removes hydrogen. Right? All right, any questions? But keep this in mind. All the other names, even if you forget, the kinase and phosphatase, they are very important, right? In eukaryotic cells, a lot of control is exerted by kinases and phosphatases. In fact, the entire cell division machinery is controlled by the activity of kinases and phosphatases. And we'll see that later on in this course. ATPase, okay? ATPases are enzymes which hydrolyze ATP, okay? So if you hydrolyze ATP, what are you going to get? ADP, yes, yeah. First hydrolysis will give you ADP, then maybe AMP. So ATPases are enzymes which break up ATP, right? And many proteins that have an energy harnessing ATPase activity as part of their function, right? Because that protein is doing some activity that requires energy. Where is it going to get that energy from? By hydrolyzing ATP, right? So that enzyme will be called an ATPase. For example, motor proteins such as myosin, okay? This is the protein that allows you to move, right? Your muscles, all right? They have to all fire together. Yeah? All the muscle cells have to fire together. Okay, they have to use up ATP, produce the energy, and that's how you and I can move, right? So an example of an ATPase is myosin, a muscle protein that is responsible for generating movement, right? Another example is membrane transport proteins such as the sodium-potassium pump, right? See, inside the cell, right? You have the cell membrane, right? The plasma membrane. You have the interior of the cell. The concentration of the sodium and potassium ions is very important, right? They have to be kept within certain tight limits. Okay, and how is that done? There is a pump in the membrane that is going to pump sodium ions across the cell membrane, okay, in the right direction. So that's why I told you, until you die, there is no rest. Okay, all your cells are working pretty hard. So because there has to be some concentration gradient that has to be maintained for some other activities, right? So there is this sodium-potassium pump which is constantly acting on your cell membrane, and making sure that the concentration of sodium and potassium inside the cell, they are held at the right levels. Okay? And obviously, if you're pumping something, you need energy. Okay? So these guys have an energy harnessing activity. That means they use ATP to get the energy that they need for pumping sodium and potassium in the right direction. And in fact, I have heard that sometimes, because if you have a real bad stomach upset, right, and you're losing fluids from the body, then the potassium sodium in your cells can be, the balance can be disrupted, and that's when sometimes people can lose consciousness, or they start talking rubbish and all that stuff happens. So it is an important function. Okay, now we move on to the mechanics of reactions between an enzyme and its substrate. Remember, the molecule that the enzyme acts on is called the substrate. Okay? So suppose you have an enzyme. As you bring in more and more substrate to act on the enzyme, initially, the enzyme is able to function more and produce more of the product. Right? Enzyme is a catalyst. So as the substrate concentration is increased, the reaction rate initially goes up as more substrate molecules are available to interact with the enzyme. Right? However, if you have a limited amount of enzyme and you just keep on, putting in more and more substrate, so beyond a certain substrate concentration, the reaction rate will saturate, since the enzyme is already functioning at its maximal speed, right? And it cannot process substrate molecules any faster. And this limiting velocity of the enzyme is called Vmax, right? And here is the diagram, right, showing you the reaction pathway. See, this is the rate of reaction. This is the concentration of the substrate, right? So as you increase the concentration of the substrate, the rate of reaction was initially 0, it starts going up, then it saturates at Vmax. Okay, this is the maximal velocity of the enzyme. Now, biologists are typically also interested in another quantity that is called Km, right? This Km, right, or it's called the Michaelis constant. This is the substrate concentration at which the enzyme is able to function at half its maximal velocity. So see, the maximal velocity of this enzyme when the substrate concentration is very high is Vmax, right? You want to find out what is the substrate concentration at which the enzyme is going to function with a velocity of 1/2 Vmax, right? And that concentration is called Km, right? These are important parameters for biologists, right? Even people in medicine and all that. Vmax and this Km for an enzyme, they are important. Why? see, again, I mean, the biologists are not engineers, okay? If it was electrical engineers, we probably say, oh, this is like a first order RC circuit. We model it with time constant and all that. No, they don't do that. They are just interested in these two numbers, Vmax and Km, okay? Now, the reason why these two numbers are important is because they will tell you, suppose you have the enzyme, the same enzyme, okay? It's used in the liver, right? It's also used in the muscle cell, okay? So where should if you have, the substrate floating around, okay? When you have excess of the substrate, where should it go? And when you have a limited amount of the substrate, where should it go? All right, that needs to be decided. That is decided by the value of the Vmax and the Km. When you have an unlimited quantity of the substrate, okay, the substrate amount of substrate available is very large, all right? And let's say you have two enzymes that are competing for that substrate. The one that has got the higher Vmax, that is the one that will get most of the substrate. Okay. When there's a lot of substrate. However, when there is a shortage of substrate, and let's say two enzymes are competing, the one which has got the smaller Km, right, that's the one that will get the substrate, right? And this is important because, see, your blood glucose levels have to be maintained within certain limits, right? So if you have, let's say you've eaten too much, right? You don't exercise, you eat too much, your blood glucose goes up, okay? So where should that glucose go from your blood, right? Where should that glucose go in your body? If it has become too high, where? It should go, no, but it shouldn't stay that long in the blood, okay? Blood glucose level has to be within certain limits, right? Otherwise, you can ask any diabetic what kind of complications they have, all right? So it has to stay within certain limits. Where should it go? Anybody? Oh, you're way ahead, okay? Yeah, I mean, triglycerides is much later, right? But to become triglycerides, where should it go first? Which organ in the body should it go to? Liver, yes, yeah. It should go to the liver, okay? Because then the liver will convert it into glycogen, store it as glycogen, and later on maybe do triglyceride synthesis, right? But the point is that if the level of glucose in the blood is high, all right, you want the excess glucose to go to the liver, not to the muscle, right? But on the other hand, if you're exercising like crazy, right, your blood glucose is dropping, you haven't even eaten, right? The supply of glucose is limited, okay? Now it needs to go to the muscle because the liver is going to store more stuff, okay? Liver does not use glucose, right? The liver does not use glucose. Liver processes glucose. Liver uses fatty acids, right? It doesn't use glucose, right? To run. So if there is an excess of glucose in your blood, that should go to the liver, right? Right? Whereas if there is a limited amount of glucose in your blood, right? Whatever little glucose you have, that should go to your muscle because you're exercising, let's say, right? So how is that carried out? Now, the liver and the muscle cells, they both have similar kind of enzymes, all right? They're called kinases. Remember, what is the function of a kinase? What does a kinase do? Yeah, go ahead. Adds a phosphate group, right? So if you are going to get energy from glucose, what is going to happen is glucose will become glucose 1-phosphate, okay? And that reaction is actually catalyzed by a kinase, right? So in the liver, there is something that is called hexokinase, right? And whereas in the muscle cell, it is glucokinase, okay? Same kind of kinase, but they have little bit different characteristics. The one in the liver, right? The one in the liver has got a higher Vmax, right? While the one in the muscle cell has got the lower Km, okay? So if the level of glucose in the blood is high, right? That means there's excess of substrate glucose, right? It will go to the place to the enzyme that has got the higher Vmax. That means it will go to the liver and get processed over there for storage as glycogen, as triglycerides and so on. On the other hand, if the substrate concentration is low, your blood glucose is falling, right? And how will you know that your blood glucose is falling? Make a guess, huh? Well, you feel all the time. Yeah, go ahead.
AMPs, you have more AMPs than ADPs, right?
Yeah, but I don't, see, I'm an ignorant man, okay? I don't understand AMPs, ADPs, okay? How do I figure out, that my blood glucose is falling, right? You become hungry, okay? That means your blood glucose is falling, right? So that's another way of saying, like, or if you want to satisfy hunger, have a soda or whatever, that'll get your glucose up, right? So So the point is that if you have a high level of glucose in the blood, when the substrate is in excess, then the substrate should be going to the enzyme that has the higher Vmax, which is there in the liver. Okay. Whereas if the substrate is a limiting factor, then it should go to the muscle. And there are lots of examples of that. That's why the biologists like to focus on these two parameters of an enzyme, Vmax and Km. So as I said, the substrate concentration at which a given enzyme can function at half its maximal velocity is called the Km for that enzyme, and it measures the affinity of the particular enzyme for the substrate. If two enzymes compete for the same substrate, then the values of their Vmaxes and Kms determine which reaction will get preference. If the substrate is limited, then the enzyme with a lower Km gets preference, while if the substrate is not a limiting factor, then the enzyme with the higher Vmax is going to dominate. Now, as I've been saying from the beginning of this chapter, right, proteins are extremely versatile molecules. They are responsible for most cellular functions. Despite their versatility, there are instances where a protein will need the help of a non-protein molecule for performing functions that would have otherwise been impossible. For example, a molecule of hemoglobin, right, that's a protein. It has got 2 alpha globin units and 2 beta globin units, but it needs the help of 4 heme groups, right? Heme is iron, right? Ring-shaped molecules that contain iron in them. Okay, that's why blood has the red color, because of iron in it. Okay, so it needs the help of 4 heme groups in order to be able to perform its function of picking up oxygen in the lungs and releasing it in the tissues. Right, so it needs a non-protein molecule. Right, the 4 heme groups that is coming from iron. Right, that's a non-protein molecule. Another example is that the signal receptor protein, right, rhodopsin, in your eye, right, in the retina, detects light by means of a small molecule that is called retinal, which is embedded in the protein. And this retinal is not a protein, right? It's a different kind of molecule. Now the cell can regulate how much of each enzyme to produce by regulating gene expression, right? We haven't gotten to that as yet, but gene expression means the activity of a gene, right? Because the genes code for proteins, right? I'll show that to you later. The gene is coding a stretch of DNA that codes for a protein, right? So a cell could regulate how much of each enzyme or each protein is being produced by controlling gene expression or the activity of genes. And we'll see how that is done in a later chapter, right? The cell can also control enzymatic activities by confining sets of enzymes to particular subcellular compartments, right? Let's say you have an enzyme, okay, but the substrate is outside, whereas the enzyme is compartmentalized. Then the reaction will not take place, right? So the cell can exert some control on whether the reaction will take place or not. An enzyme can also change its activity in response to other molecules that it encounters in the cell, right? And we'll talk about this a little bit later. And an example of an enzyme changing its activity in response to the presence of other molecules is feedback inhibition. This is nothing new to engineers, right? But in the context of biology, you know, it's something probably that may have puzzled them before. So in feedback inhibition, what is happening? Like you have W is getting converted to X, some molecule W getting converted to X, then getting converted to Y, then getting converted to Z. Each of these conversion steps is being catalyzed by some enzyme, like this one by enzyme E1, this one by enzyme E2, by enzyme E3, right? However, the product of the last step, right, this Z is producing something that is going and negatively regulating the enzyme E1. That means if too much of this guy, if this reaction goes too much in the forward direction, a lot of this stuff will produce, and that'll put the brakes, okay? So there is feedback inhibition. So in feedback inhibition, X produces Y, which produces Z. However, this Z then goes and blocks the production of X. And this is an example of a negative feedback loop, right? For those of you that have taken 420 or control systems, you know that you have these kind of loops all over the place, right? So in general, so this is just an example of a single feedback loop, right? But in general, in the body, there will be multiple, or in the cell, there will be multi-loop feedbacks, right? Yes, sir. Go ahead.
E1, enzyme E1 will be affected by...
Yes, by Z. So Z can come and affect the enzyme E1, okay? Maybe Z also wants E1 and takes it away, doesn't let it act on W, right? It can happen that way. And there are lots and lots of examples of that. And we will see some of those even later. Okay. Now, another example of negative feedback is when there is a rise in ADP in the cell, right? Again, ADP is the adenosine diphosphate, right? So that's a low energy state. So when there is a rise of ADP in the cell, which is indicative of a low energy state, several enzymes involved in the oxidation of sugar molecules are activated, right? Because you need sugar, you need to oxidize glucose in order to produce more energy, right? So when these enzymes are activated, this will stimulate more glucose to be burned, and the cell will convert ADP to ATP, right? And this lowers the level of ADP in the cell and will restore the desired energy level, right? That's if everything is working okay, all right? If you're diabetic or something like that, then there's a problem, right? Because even you might be feeling hungry, your energy level is low, even then there is sugar staying in your blood, okay? It's not going to the cells to power them, right? That's the problem. Yeah.
With diabetic people, they do not convert ADP into ATP as they should. That's the
No, there is something called AMP activated protein kinase, right? Adenosine monophosphate AMPK, AMP activated protein kinase, and one of the drugs that is used for treating diabetes, all right, it's called metformin, all right, or I mean some of you might know of it as glucophage or something like that, right? That one actually is an AMP kinase activator, okay? So if you give that to a diabetic person, then their AMP kinase will get activated, right? And that will lower the blood sugar, right? So it's all at the cellular level. I mean, we might see it at a higher level, but really it's something that is happening at the level of the cell, function of the cell. Yeah. I mean, and not only that, in the case of diabetics, all right, there are two types of diabetes. Okay, one is called type 1 diabetes, all right. The other one is type 2 diabetes, all right. Type 1 diabetes is basically kids typically have that, right? Huh?
it's the one that's like genetic.
Yeah, it's not the one that doesn't develop it. Basically, in that case, the body does not produce enough insulin. Okay, so somehow the beta cells in the pancreas are damaged, maybe because of virus infection or whatever. Right? Body does not produce insulin. Now, what does insulin do? Well, insulin goes and binds to cells. It increases the number of glucose transporters because the glucose from the blood has to enter the cells. Okay, before all the reactions will run. Right? So if insulin, so if you eat, okay, your blood glucose goes up, right? Insulin is secreted. Insulin is going to go and increase the number of glucose transporters on the cell, so the glucose from the blood is going to flow inside the cell and is going to be used up over there, okay? So then your glucose comes back to the normal level. That's for normal people, right? But if somebody is diabetic, right, they've eaten, the glucose level stays there, right? It's not coming inside because the cell probably doesn't even know because its AMP kinase impaired, right? It doesn't even know that more glucose needs to go in, right? So when there is a rise in ADP in the cell, which is indicative of a low energy state, or even cyclic AMP, right, that I talked about, adenosine monophosphate, okay, that's a signaling molecule, okay, that is indicating to the cell that, hey, you better start firing on all cylinders, So several enzymes involved in the oxidation of sugar molecules are activated. This will stimulate the conversion of ADP to ATP. This will lower the level of ADP in the cell and will restore the desired energy level, okay. So it's all like a feedback system. So if you have a feedback system and something is messed up, that's when you're in trouble. Even in cancer it's the same, okay. There is a balance, you go and mess it up, and then there is a problem. So next I want to talk about a special class of proteins that are called allosteric proteins, right. Now in feedback inhibition, right, when you're producing some product and then that is slowing down the reaction, oftentimes it is found that the regulatory molecule has got a shape that is totally different from the shape of the substrate of the enzyme, right? Because see, if you look at this, okay, this picture here, you might think, okay, this enzyme E1 is acting on W, right? Now this guy Z is negatively regulating this reaction. How? Because Z is probably preferentially binding to E1. Okay, in the location on E1 where W was binding, this guy got in and now W cannot bind, so the reaction is not going forward. That's the case in some situations, but in some other situations, people have done analysis and the shape of W, the molecular shape of W, is completely different from that of W. Molecular shape of Z is completely different from the molecule of W. So how does the reaction stop in that case? Okay, that's the question, right? Yeah.
Here is regular molecule.
Here, what is regular molecule? No, Z is the regulatory molecule. Yes, yeah. Regulatory molecule means any molecule that is regulating the activity, so. Okay. Z is the product of that reaction, but it's also functioning as a regulatory molecule. So in feedback inhibition, oftentimes it is found that the regulatory molecule has got a shape that is totally different from the shape of the substrate of the enzyme, right? And in this case, the feedback inhibition is not caused by the regulatory molecule binding preferentially to the same binding site on the enzyme. Instead, what usually happens in these cases is that the enzyme has got two binding sites, one for the substrate and the other for the regulatory molecule. And such proteins are called allosteric proteins. So when the regulatory molecule comes and binds the protein at one of the sites, okay, that slightly changes the shape of the protein, right? And as a result, the original molecule cannot bind to the other site. So that's how allosteric proteins function. And in fact, these kinase and phosphatase, okay, I talked about that. I said that they play an important role in switching reactions on and off. And the way it happens is when a phosphate group comes and binds, right? Phosphate has got two negative charges, right? So in a protein, you suddenly have two negative charges appearing. That means the shape of the protein has changed, okay? Somewhat, it has changed somewhat. So the original binding site might have changed. So the molecule which was supposed to come and bind over there cannot bind anymore. So eukaryotic cells commonly... Okay, just wait. Maybe I missed something. No, I did not. So let's look at regulation of protein function by a change in the conformation of the protein. Eukaryotic cells commonly regulate a protein's function by adding a phosphate group covalently to one of its amino acid side chains. Now, because each phosphate group carries two negative charges, right? The enzyme catalyzed addition of a phosphate group to a protein can cause a major conformational change, right? Because you have to put in two negative charges. Remember, the three-dimensional conformation of a protein is determined by the interaction of many different factors, okay? If you throw in like two negative charges over there, you put in an additional factor, that might alter the shape of the protein, right? And therefore, it can no longer perform its original function, right? So to restore the original function, you can remove the phosphate group by a second enzyme. The addition of a phosphate is carried out by a kinase, alright? The removal of a phosphate is carried out by a phosphatase. So removal of the phosphate group by a second enzyme, which is usually called a phosphatase, returns the protein to its original conformation and restores its initial activity. So it's like a switch that can turn on and off. Add a phosphate, maybe it turned on, you take off the phosphate, it turned off, and vice versa. And in fact, there are lots of examples of those kind of proteins. So in eukaryotic cells, protein kinases and protein phosphatases play an important role in switching proteins from inactive to active conformations and vice versa. For example, proteins undergo dramatic conformational changes when they bind GTP. Just like you have ATP, there is a GTP because there is AGCT, alright? GTP is guanosine triphosphate. The protein that is bound to GTP is active, right? In fact, it is hyperactive. It's like supercharged. It's like a hyperactive kid, right? Then what happens is it becomes hyperactive, but after some time, the GTP loses that one phosphate group, becomes GTP, right? And the protein becomes inactive. After some time, it gets rid of that GTP, and then it's still inactive. But again, when it is bound by GTP, it's again active, right? So after a while, the bound GTP is released, but the protein remains inactive until it binds the GTP again. And there are lots of examples of proteins that are activated by GTP, right? In fact, RAS is an oncoprotein, right? That is active. So normally in the case of RAS, okay, RAS is something that can cause cancer, okay? It plays a role in the control of cell division. So it's a GTP binding protein. So in the normal situation, what is going to happen is that RAS is going to get bound by GTP, all right? It's activated, it starts functioning. But then the GTP gets hydrolyzed to GTP, all right? And the RAS loses its activity, right? But in the case of cancer patients, what is going to happen is that the GTP stays permanently as GTP. It doesn't get, so RAS would have lost its hydrolyzing activity and it's permanently bound to GTP, so it is really active, okay? So it can really wreak havoc, right? So next I wanted to point out that proteins can also produce movement, right? So, and motor proteins, they can produce directed movement in cells by coupling the hydrolysis of ATP to conformational changes in the protein. So, and to see that, let's go to the picture, right? So a motor protein that can walk just like you and me, okay? So this is a protein, right? Schematic diagram for a protein, so then it's going to move from here, okay? So it's trying to walk. then it's bound by ATP, A and then 3 phosphates, right? Then with the next step, right, when this thing moves over here, right, the ATP is hydrolyzed, right? So from ATP, it will become ADP, all right, 2 phosphates and A and 2 phosphates and 1 phosphate ion is released. So in the next step, all right, you can see that once it's released, it's back to the original conformation. Like it was here, it has moved, all right? Now what has ATP hydrolysis got to do with this? Because normally if you didn't have the ATP hydrolysis, it will be like what is called a random walk, right? It can move either this direction, that direction, okay? By coupling this with a reaction that is energetically favorable, okay, you have hydrolyzed the ATP to get from here to here, right? That's a step that is not easily reversed, okay? The reverse reaction, that is adding a phosphate group to ADP to produce ATP, is energetically unfavorable. It's not going to take place, okay? So the result is that by coupling this movement with the hydrolysis of ATP, right, you're producing directed movement, right? You're going to go only in this direction. It's not going to spontaneously spring back in the opposite direction, right? So that's how a protein can walk, and that's how proteins walk in, like when they produce DNA, RNA and so on. Okay? They need to move along the DNA strand to make a copy of the DNA strand. So that's how the protein walks, because the energy is needed for that, right? So again, I mean, this is a textual description of whatever I showed you in that diagram. So motor proteins can produce directed movement in cells by coupling the hydrolysis of ATP to conformational changes in the protein. In the absence of ATP hydrolysis, the protein would move back and forth in different directions, generating random movement, right? And for signal processing people here, or stochastic process, that's a random walk, right? So by coupling the movement to ATP hydrolysis, which is an energetically favorable reaction, the movement steps are made irreversible, thereby producing directed movement. And this is the mechanism by which DNA and RNA polymerases move along a template DNA strand. And we'll talk more about this later, once we have covered DNA in some detail. That's coming up in the next chapter. All right, any questions? All right, we're finally at DNA. Okay, so we finished the chapter on proteins. That was quite a long chapter, all right? So if there are any questions about proteins, right? We covered a lot, and I mean, if you're a biology major, they'll give you like hands-on experience, how to run all those electrophoresis, then all the column chromatography and so on, okay? But now we have arrived at the chapter on DNA. DNA basically is the abbreviation for deoxyribonucleic acid, right? So why is it called deoxyribonucleic acid? Because the sugar in there is deoxyribose, right? So now we know, right? When we started this class, we didn't know what a sugar was. Now we know what deoxyribose is. Right? That's the sugar in DNA. And it's a nucleic acid because it is made up of nucleotide units that are kind of linked together. So DNA or deoxyribonucleic acid is the molecule that is used in cells to store genetic information. Right? And today it's like second nature. Even the person who knows nothing about DNA will say it's in my genes. Okay? It's not my fault, it's in my genes. Right? But, as I will discuss in this chapter, that until about 70 years ago, people did not even know that DNA was the carrier of genetic information, right? So that's only 70 years old. Okay? In 1944, there was an experiment that was done to demonstrate conclusively that the information that is transmitted from one cell to the other and from generation to generation at cell division is basically contained in the DNA. Now the genetic information must be passed on from a cell to its daughter cells at cell division, because you want to produce two identical copies of the original cell. Then this information is passed on from generation to generation of organisms through the reproductive cells, right? Like you inherit traits from your parents, your grandparents, your great ancestors, okay, all that is being transmitted through DNA. Okay. What are genes? Right? A gene is roughly a stretch of DNA that codes for one protein. Right? Genes are the information containing elements which determine the characteristics of a species as a whole and also of the individuals within it. And the natural questions that come up are, what is the chemical composition of the genes? What are genes made of? Right? And what is the mechanism by which these genes can store this information, which can be replicated in an almost unlimited fashion, okay, across thousands of years, right? From your parents, your grandparents, or whatever, across so many, if you're looking at an individual human being, probably you have about 100 trillion cells, okay? So that same information is being communicated across so many cell divisions, in a fairly faithful way, because otherwise you and I would not be here. Okay? So these are natural questions that come up. What is the molecule that is capable of storing this information and also transmitting it with minimum error? Some of you are in communications, like all those error correcting codes and all that. DNA already has that. Okay, and the rate is the rate of error is very, very low. Now, by the early 20th century, right, the biologists knew that genes were carried on chromosomes, right? Because they can see when a cell is dividing, then the DNA and the protein, okay, that forms the chromosomes, that is organized DNA, that will condense, right? And they could see under a microscope. So they knew that genes were carried on chromosomes, right? Now, biochemical analysis of the chromosomes reveal that they are made of DNA and protein. So if you analyze chromosomes, right, using chemicals, right, biochemical analysis, you find that they have DNA, right, there's nucleotide units, and then they also have protein. Now, regarding DNA, okay, what are the 4 different units that you can have in DNA? A, G, C, T, right? So if you look at a string of DNA, it basically is a sequence of letters A, G, C, T, right? So not that much variety. At every position, there are only 4 possibilities, okay? Whereas if you look at a protein, right, it's made up of amino acids. How many amino acids are there in nature? 20. Okay, so it's a much, much richer, molecule in terms of the number of combinations that you can generate, right? That's why people initially thought that proteins must be the carrier of genetic information, right, and not DNA. So that was the initial thinking, because DNA is such a simple molecule, that it's just got 4 letters. How can that be the one? It would be probably the one that has 20. Possibilities at each location. So this notion was dispelled by an experiment that was conducted in the year 1944 to show that DNA is the carrier of genetic information. And this is how the experiment went. There were 2 closely related strains of the bacterium Streptococcus pneumonia were used, right? You want to make a guess what this bacterium will cause? What disease is this bacterium going to cause? It's Streptococcus pneumonia. You cannot take a guess. What is the disease that this bacterium will cause? Huh? Pneumonia, yeah. It causes pneumonia. Right, that's why it's Streptococcus pneumonia, right? Anyway, there were 2 strains of this bacterium. The R strain, okay, it's called R because it looks rough under a microscope, is not pathogenic. It's not harmful, right? And doesn't cause pneumonia when injected into animals, right? So if you take a bunch of rats, you inject them with this version, the R strain, the animals are doing fine. They don't die. The S strain, it looks smooth under the microscope. It's pathogenic and it causes pneumonia when injected into animals, resulting in death, and the animals die. Okay. So what they did is basically they went ahead and killed, right? See, you have this S strain, okay? Go ahead and kill it, boil it in water or something like that, right? Kill the S strain bacteria, right? And then use that soup to grow some R strain bacterium, right, on there. The R strain is normally harmless, but you're growing it on heat-killed S strain bacteria, right? And then when they took this R strain that was being grown on the heat-killed S strain, and they injected it into animals, right, into rats, those rats died, okay? So that tells you that somehow that R strain, okay, by feeding on the whatever was left over from the heat-killed S strain, it got transformed to the S strain, okay? And then, so somehow the R strain got transformed to the S strain, okay? Then they went ahead and did the analysis, okay, for the R strain one, right? Let's go and, the R strain that was grown on the heat-killed S strain, let's go and analyze all the molecules, all right, like lipids and, carbohydrates, proteins, DNA, everything, okay, they analyzed. All the other molecules were found to have stayed the same. They were still of the R strain type. Only the DNA, right? Only the DNA had been changed from the R strain to the S strain when these guys were made to feed on the heat-killed S strain, right? So the conclusion is that the carrier of genetic information is the DNA, right? So I'll repeat this again, after I've explained. So here, live R strain cells were grown in the presence of heat-killed S strain cells. The animals that were injected with these cultured R strain cells died, leading to the conclusion that the R strain cells had been transformed to S strain cells. So the conclusion is that the molecules that carry heritable information are present in the heat-killed S-strain cells. Okay? Because if they were not, then these R-strain cells would not have been altered, right? So the next step is to go and get the cell-free extract from these transformed R-strain cells was fractionated into classes of molecules, okay? And we have already covered procedures to do that, right? I mean, we talked about all those sedimentation, centrifugation, and things like that. You separate it out. It was found that the resulting RNA, proteins, lipids, and carbohydrates, they were all found to be of the original type, of the R-strain type. The only thing that changed was the DNA. The DNA was found to have been transformed to the S-strain type. So then the conclusion was that DNA carries the heritable information, right? DNA is the carrier of genetic information. Right? So now we move on to, okay, any questions so far? Now we move on to the structure of DNA, right? The structure of DNA was discovered by James Watson and Francis Crick in 1953, right? And this discovery actually eluded people for many, many years. These guys got the solution in 1953 and they got a Nobel Prize, okay? However, there were several prominent people that were struggling with this structure, including Linus Pauling, right? The famous guy at Caltech, okay, who got 2 Nobel Prizes. He also worked on this problem. But I think he made a mistake in calculating some bond length somewhere, so everything didn't fit, okay? So he didn't get it. These two guys were quite young at that time, right? And they were working on this, and I'm told that they had built a model using, sticks and chewing gum, right, to see how everything would fit in, okay? But they didn't quite have the entire picture. But there was this lady, right, who had she or her husband used to work on X-rays, okay? But they were looking at it from a different point of view. They were trying to take X-ray pictures. And these guys, somehow they managed to see that picture, okay, and then it clicked and then they got it. And she unfortunately died before the Nobel Prize was awarded and they don't give the Nobel Prize to a dead person, so, and maybe it was not even that publicized at that time, but, so, even famous people, they do unethical things, yeah. You have a question? But so this discovery was made in 1953. And we have already talked about the DNA base, all right? It can be a purine or a pyrimidine, all right? Remember the 9-member ring or 6-member ring. That connected to a 5-carbon sugar. In the case of DNA, it is deoxyribose, okay? So that's the sugar, right? So nucleotide, sugar, and then the phosphate. And remember, we had that 5' end and the 3' end, right? On the sugar, there was 1', 2', 3', 4', 5', right? Now you take these units, okay? So this is like the 5' end, right? Then this is like the 3' end, right? So you take so here you have the base, then you have the sugar phosphate, right? And the detailed chemical structure I already discussed in chapter 2, right? So you now go and string these units together, you get a single strand of DNA. So again, it has a polarity, 5' at one end, 3' at the other. These things are linked to each other by phosphodiester bonds, right? Because this was already, here there is already a single phosphoester bond linking this sugar to this phosphate. And when you link it to the next nucleotide, it's ester bond on both sides, so it's a phosphodiester bond, right? So that gives you a single strand of DNA. However, DNA always occurs in nature as a double-stranded molecule, right? So if you have one strand like this, the other strand like this, right? There is a complementary base pairing property. A always pairs with T. Look, over here, A is always pairing with T through 2 hydrogen bonds, okay? G pairs with C with 3 hydrogen bonds, okay? And this business of A pairing with T and G pairing with C is going to make sure that if you think of this as a ladder, all the rungs of the ladder have the same length, okay? This was the big thing in figuring out the structure of DNA, right? So there is a definite polarity. This is the 5' end, this is the 3' end. The other strand is running here 5' to 3'. Okay, and why do we have prime instead of the regular numbers? Anybody remember? Yes, because the regular numbers have been used up for the bases. So the sugar is using the primes, okay? Now on top of that, okay, so this is like the ladder. So the double-stranded DNA, it's like a ladder-like structure. And then this thing, it spirals around itself like a spiraling staircase, right? And this produces the double helix, right? The famous double helix that you may have seen in all kinds of magazines, including Time, People, Newsweek, whatever, right? You do not have to be like a molecular biologist or taking, genomic signal processing engineering application to know about that. You would have seen it somewhere, if your eyes were open, all right, in the last especially 50 years after this, like 53 was the year it was discovered. So in 2003 there was a special issue, I think, of Time, 50 years of the double helix and so on. Okay, so this is a big thing because this has really changed the course of biology, medicine, and a lot of other things. Okay, the discovery of this, because once they figured out this structure, right? Now they know how it replicates, because the two strands can separate, right? You can use one of the strands as a template, right? And we engineers, we know about it. Okay, like you have 2's complement. Okay, wherever there's 1, you put a 0, 0, 1, and so on. Okay, so you can figure out if one of the strands is given to you, you can figure out the other strand. Moreover, since we have double-stranded DNA, see, if something gets messed up, right? Let's say something got messed up. Then there are mechanisms in your body that can detect that cut out the mess up, look at what is on the other strand, right? And produce the correction here. That's your error correcting codes, right? Okay, for communication people. All that is inbuilt in the cell. If that was not there, you would be dead. Okay, you're not. I mean, many of you are probably in your 20s. Okay, you would not reach your 20s. Okay, you'd be dead long time back. Maybe even before you reach the age of 2. All right. So these things correct. Every time you step out into the sun, some of this got messed up, right? So start staying indoors all the time. No, there is a saying that they say that if you live to be long enough, all right? I mean, if you live long enough, you will ultimately die of cancer, okay? Because some of this stuff will get messed up, and your correction mechanisms also, they slow down with age. Okay, it's not that you just look old. Okay, as you're growing older, even your DNA repair mechanisms slow down. Okay, so that's an unfortunate fact of aging, right? So this is the structure of DNA that was discovered in 1953 by Watson and Crick. Yes, sir. Go ahead.
So last trend is a map.
What?
Another one trend is map of
No, the complements, not maps. Like if you have on this string, right, a G at any location, let's say G here, it will always pair up with a C on the other strand. Okay? If you have an A here, it will always pair up with T. Unless something is messed up, it should pair up with T. Okay? And then the length is the same all throughout this ladder, and then it rotates around itself like a spiraling staircase. Yeah.
Why C's and G's have like 3 links?
Yeah, because G and C are linked together by 3 hydrogen bonds. Okay. Well, I'm not a chemist. You look at the structure, right? Yeah, see, that's the reason why in the 1950s and all so many people struggled, right? I mean, they struggle for, I don't know the answer to your question, but I'm sure you'll find it, because this has been around for almost 60 years, more than 60 years, right? I mean, 1953. So, I mean, it is well documented in their papers. I'm sure of that. Okay, but it's just I didn't, and you'll see why it would not form. They're good questions, and I'm sure that they have been answered. Okay, but it is 3 hydrogen bonds, G and C, right? 2 hydrogen bonds will link A and T. Okay. Furthermore, one more thing to note from here: if the 2 strands need to come apart, okay, where should those openings take place? Which one will be easier to break? AT. Yeah. So usually those are called replication origins. We'll get to that. They're usually where you have AT, because those are easier to break. Right? All right, any other questions? So let me just continue. So I'll quickly finish up this part and then we'll stop. So a DNA molecule consists of 2 complementary chains of nucleotides. 4 different nucleotide bases make up DNA: adenine, guanine, cytosine, and thymine. A pairs with T through 2 hydrogen bonds, while G pairs with C through 3 hydrogen bonds. And again, if you want to delve deeper into the reason, it is there in the literature, in the public. Even Google might have it. Sorry, not. What is it? Is it Google, or where do you look? Wikipedia, whatever, they'll have that information. The pairing of A with T and G with C ensures that each pair is of similar weight, thus holding the sugar phosphate backbones an equal distance apart along the molecule. In addition, the 2 sugar phosphate backbones will wind around each other to form this famous double helix that you see all over the place. So DNA encodes information in the order or sequence of the nucleotides along each strand. Each base A, G, C, or T can be considered as a letter in a 4-letter alphabet that is used to spell out biological messages in the chemical structure of the DNA. Organisms differ from one another because their respective DNA molecules have different nucleotide sequences. So if you look at a human cell, there are 3 billion of those letters. Okay, The initial attempt to sequence that, to find out all those, took many years, and as Dr. Johnson said, it cost like billions of dollars. Okay, today in his lab he can do something in 24 hours for, I don't know, $1,000 or something like that. He can do it. So the linear sequence of nucleotides in a gene spells out the linear sequence of amino acids in a protein. And the exact correspondence between the 4-letter nucleotide alphabet of DNA and the 20-letter amino acid alphabet of protein, which is called the genetic code, will be discussed in the next chapter. Here we are going to focus on the mechanisms by which DNA is replicated, possible errors that occur during DNA replication, and the mechanisms that are used to correct them. Right? So that will be the focus of the rest of the chapter, so I will start talking about it on Wednesday. Okay?